Now let's learn about mass spectrometry. Mass spectrometry is a powerful analytical technique used to quantify known materials, to identify unknown compounds within a sample, and to elucidate the structure and chemical properties of different molecules. The complete process involves the conversion of the sample into gaseous ions and with or without fragmentation, which are then characterized by their mass to charge ratios and relative abundances. This technique is basically studied the effect of ionizing energy on molecules. This depends upon chemical reactions in the gas phase in which sample molecules are consumed during the formation of ionic and neutral species. So when we look at the basic principle, a mass spectrometer generates multiple ions from the sample under investigation. It then separates them according to their specific mass and then records the relative abundance of each ion type. The first step in the mass spectrometer analysis of compounds is the production of gas phase ions of the compound, basically by electron ionization. This molecular ion undergoes fragmentation. Each primary product ion derived from the molecular ion in turn undergoes fragmentation, and so on. The ions are separated in the mass fragmentation and ions are separated in the mass spectrometer according to their mass to charge ratio and are detected in proportion to their abundance. A mass spectrum of the molecule is then produced. It displays the result in the form of plot of ion abundance versus mass to charge ratio. Ions provide information concerning the nature and structure of their precursor molecule. In the spectrum of pure compound, molecular ions of molecule. In the spectrum of pure compound, the molecular ion, if present, appears at the highest value of the m by z, followed by ions containing heaviest isotopes, and gives the molecular mass of the compound. Next, compounds and components. The instrument consists of three major components like ion source for producing gaseous ions from the substance being studied and analyzer for resolving ions into their characteristic mass components according to their mass to charge ratio. Next, a detector system is needed for detecting the ions and recording the relative abundance of each of the resolved ionic species. In addition, a sample introduction system is necessary to admit the samples to studies to the ion source while maintaining the vacuum region remains on the reactant, and the computer is required to control the instrument, manipulate data, and compare the spectra to the reference libraries. With all the above components, a mass spectrometer should always perform the following procedures: produce ions from the sample in the ionization source, separate these ions according to their mass to charge ratio in the mass analyzer, eventually fragment the selected ions and analyze the fragments in a second analyzer. First, produce ions from the sample in the ionization source, separate these ions according to their mass to charge ratio in the mass analyzer, then eventually fragment the selected ions and analyze the fragments in a second analyzer. Detect the ions emerging from the last analyzer and measure their abundance with the detector that converts the ions into electrical signals. Process the signals from the detector that are transmitted to the computer and control the instrument using feedback. Analysis of biomolecules using mass spectrometry. Mass spectrometry is fast becoming an indispensable field for analyzing biomolecules. Till the 1970s, the only analytical technique which provide similar information were electrophoretic chromatographic or ultracentrifugation method. The results were not absolute as they were based on characteristics other than the molecular weight. This is only possible for knowing the exact molecular weight of a macromolecular substance obtained its calculation based on its chemical structure. The development of deabsorption ionization methods based on the emission of pre-existing ions such as plasma desorption, fast atom bombardment, or laser desorption allowed the application of mass spectrometry for analyzing complex biomolecules. Analysis of glycans. Oligosaccharides are molecules formed by the association of several monosaccharides linked through glycosidic bonds. The determination of the complete structure of oligosaccharides is more complex than that of proteins or oligosaccharides. It involves the determination of additional components as a consequence of the isomeric nature of monosaccharides and their capacity to form linear or branched oligosaccharides. Knowing the structure of the oligosaccharide requires not only the determination of its monosaccharide sequence and its branching patterns, but also the isomer position and the anomeric configuration of each of its glycosidic bonds. Advances in glycobiology involves a comprehensive study of structure. Advances of glycobiology involves a comprehensive study of structure, biosynthesis, and biology of sugars and saccharides. Mass spectrometry is engineering emerging as an enabling technology in the field of glycobiology. Analysis of lipids. Lipids are made up of many classes of different molecules which are soluble in organic solvents. Lipidomics, a major part of metabolomics. Molecular weight, element composition, and nature of the lipid structure are analyzed. Analysis of proteins and peptides. Proteins and peptides are linear polymers made up of combinations of the 20 amino acids linked by peptide bonds. Proteins undergo several post-translational modifications extending the range of their function via such modifications. The term proteomics refers to the analysis of complete protein content in a living system. Introducing co- and pro-translational modified proteins and additional alternatively spliced variants. Mass spectrometry has now become a crucial technique for almost all proteomics experiments. It allows precise determination of the molecular mass of peptides as well as their sequences. This information can very well be used for protein identification, de novo sequencing, and identification of post-translational modifications. Now, analysis of oligonucleotides. Oligonucleotides, DNA or RNA, are linear polymers of nucleotides. These are composed of a nitrogenous base, a ribose, sugar, and a phosphate group. Oligosaccharides may undergo several natural covalent modifications which are commonly present in tRNA and rRNA, or unnatural ones resulting from reactions with exogenous compounds. Mass spectrometry plays an important role in the identification of these modifications and determining their structure as well as their position in the oligonucleotide. It not only allows for determination of the molecular weight of oligonucleotides but also in a direct or indirect manner the determination of their sequence. Software for mass spectrometry data analysis. SimGlycan predicts the structure and other information about glycans and glycopeptides from the mass spectrometry data required by mass spectrometry, facilitating glycosylation and post-translational modification studies. SimGlycan accepts the experimental MS profiles of both the glycopeptides and released glycans, matches them with its own database, and generates a list of probable sequences and structures. The software also accepts multi-stage mass spectrometry data analysis, which enables structural elucidation and identification of fragmentation pathways. SimLipid is also an innovative lipid characterization tool which enables structural elucidation of unknown lipids using MS data. The software analyzes lipid mass spectrometric data for characterizing and profiling lipids. SimLipid can also annotate mass spectra with the lipid structure identified using abbreviations. Let's have a quick recap about spectroscopy. Spectroscopy pertains to the dispersion of an object's light into its component colors, energies. By performing this dissection and analysis of an object's light Astronomers can infer the physical properties of that object, such as temperature, mass, luminosity, and composition. But before we hurtle headlong into the wild and woofy field of spectroscopy, we need to try to answer some seemingly simple questions, such that what is light and how does it behave? These questions may seem simple to you, but they have presented some of the most difficult conceptual challenges that the long history of physics. It has only been in this century, with the creation of quantum mechanics, that we have gained a qualitative and quantitative understanding of how light and atoms work. You see, the questions we pose are not always easy, but to understand and solve them, we unlock a new way of looking at our universe. The nature of light. To understand the process in astronomy that generates light, we must realize first that light acts like a wave. Like light has particle like properties too, so it is actually quite a twisted beast, which is why it took so many years to figure out. But right now, let's just explore light as a wave. Picture yourself wading around on an ocean beach for a moment and watch the many water waves sweeping past you. Waves are disturbances, ripples on their water, and they possess a certain height, amplitude, with a certain number of waves rushing past you every minute, the frequency, and all moving at a characteristic speed across the water, the wave speed. Notice the distance between successive waves. This is called wavelength. Keeping this analogy in mind, let's leave the ocean beach for a while and think about light like a wave. The wave speed of light, wave is simply the speed of light, and different wavelengths of light manifest themselves as different colors. The energy of light wave is inversely proportional to its wavelength. In other words, low energy waves have long wavelengths and high energy light waves have short wavelengths. The electromagnetic spectrum. Physics classify light waves by their energies like wavelengths, labeled an increasing energy. We might draw the entire electromagnetic spectrum as given. First, we will be representing the radio waves, infrared, visible light, ultraviolet, X-rays, and gamma rays. Gamma rays have the shortest wavelength but highest energy, but radio waves have long wavelength but low energy. The electromagnetic spectrum. Notice how small the visible region of the spectrum is compared to the entire range of wavelengths. Notice that radio, TV, and microwave signals are all light waves. They simply lie in wavelengths, energies that your eyes doesn't respond to. On the other end of the scale, beware the light energy UV, X-ray, and gamma ray photons. Each one carries a lot of energy compared to other visible and radio wave particles. They are the reasons you should wear sunblock, for example. When we look at the universe in a different light, non-visible wavelengths, we probe different kinds of physical conditions and we can see how new kinds of objects. For example, high energy gamma ray and X-ray telescopes tend to see the most energetic dynamos in the cosmos, such as active galaxies, the remnants from massive dying stars, accretion of matter around black holes, and so forth. Visible light telescopes best probe light produced by stars. Longer wavelength telescopes best probe dark, cool, obscured structures in the universe, dusty star forming regions, dark, cold molecular clouds, the primordial radiation emitted by the formation of universe shortly after the Big Bang. Only through studying astronomical objects in many different wavelengths as astronomers able to piece together a coherent, comprehensive picture of how the universe works. General types of spectra. Typically, one can observe two distinct classes of spectra, continuous and discrete. Continuous spectra and discrete spectra. For a continuous spectra, the light is composed of a wide continuous range of colors or energies. With the discrete spectra, one sees only bright or dark lines at very distinct and sharply defined colors. As we discover shortly, discrete spectra with bright lines is called emission spectra, and those with dark lines are called absorption spectra. Dark lines are called absorption spectra. Next, we will be learning about the continuous spectra. Continuous spectra arise from dense gases or solid objects which radiate their heat away from the production of light. Such objects emit light over a broad range of wavelengths, thus the apparent spectrum seems smooth and continuous. Stars emit light in a predominantly but not completely continuous spectrum. Other examples of such objects are incandescent light bulbs, electric cooking stove burners, flames, cooling air, fire lamps, etc. Yes, you're right this minute. Are emitting a continuous spectrum, but the light wave you are emitting are not visible. They lie an infrared wavelength, lower energies and lower wavelengths than even red light. If you had infrared sensitive eyes, you could see people by the continuous radiation they emit. So, discrete spectra. Discrete spectra are the observable result of the physics of atoms. There are two types of discrete spectra, emission and absorption. Let's try to understand what are these two types of discrete spectra. Emission line spectra. Unlike a continuous spectrum source which can have any energy it wants, all you have to do is change the temperature. The electron clouds surrounding the nuclei of atoms can have only very specific energies directed by quantum mechanics. Each element on the periodic table has its own set of possible energy levels, and their expressions, the levels are distinct and identifiable. Atoms will also tend to settle to have lower energy levels in the spectroscopist's lingo. This is called the ground state. This means that an excited atom in higher energy must dump some energy. This way, an atom dumps that energy by emitting a wave of light. What that exact energy? In the diagram, a hydrogen atom at the second energy level of the first giving off a wave of light with an energy equal to the difference of energy between level 2 and 1. This energy corresponds to a specific color or wavelength of light, and thus we see a bright line in that exact wavelength. An emission spectrum is born. An excited hydrogen atom relaxes from level 2 to level 1, yielding a photon. This results in the bright emission line. Tiny changes of energy in an atom generate photons with small energies and long wavelengths, such as radio waves. Similarly, large changes of energy in an atom will mean that high energy short wavelength photons are emitted. Now let's learn about the absorption line spectra. On the other hand, what would happen if we tried to reverse this process? That is, what would happen if we fired the special photon back into a ground state atom? That's right, an atom could absorb that specifically energetic photon and would become excited, jumping from the ground state to the higher energy level. If a star with a continuous spectrum is shining upon an atom, the wavelengths corresponding to possible energy transitions within that atom will be absorbed, and therefore an observer will not see them. In this way, a dark line absorption spectrum is born as shown below. Now let's learn how does a spectrometer work. Many people know how a telescope works, but relatively few have much experience with the innards of spectrometer. So let's take apart the astronomy camp spectrometer to see how it works. Keep in mind that there are many optical designs of spectrometers as there are optical designs for telescopes, and that this is but the one of the simplest. Nevertheless, it puts out the salient features of most optical spectrometers: telescope axis, optical fibers, guide optics, and slit. Along with that, collimator and rating will be there. It all starts with the telescope light beam entering the spectrometer. The focal point of the telescope beam is brought to the slit of the spectrometer. This slit is what it's ultimately imaged in the detector. In the case of a camp spectrometer, the slit is arranged in an angle and the slit surrounding the slivered so that the portion of the telescope beam not passing through the slit can be routed instead of an eyepiece for easy telescope guiding. The light passing through the slit then is reflected off a collimating mirror which paralyzes the beam of light before sending it off to the diffraction grating. This optical element disperses the parallel beams of light into their component colors or wavelengths and energies. Each each different wavelength comes off of the grating at a slightly different angle. So now we have an image of a slit that is spread out like a rainbow by color. This new color dispersed beam of light is then focused and imaged in the detector by the camera lens. A 335 mm camera is the detector in this diagram, but in a camp we typically use the eyepiece of a CCD array. So now let's put all this together to make a spectrometer. If the sample compound does not absorb light of a given wavelength, I is equal to I0. However, if the sample compound absorb light when I is less than I0, and this difference may be plotted on a graph versus wavelength as shown in the light, absorption may be represented as transmittance or absorbance. If no absorption has occurred, T is equal to 1 and A is equal to 0. Most spectrometers simply absorbance on the vertical axis, and the commonly observed range is from 0 to 2. The wavelength of maximum absorbance is a characteristic value designated as lambda max. Different compounds may have very different absorption maxima and absorbances. Intensely absorbing compounds may be examined in dilute solution so that significant light energy is received by the detector, and this requires the use of completely transparent solvents. The most commonly used solvent are water, ethanol, hexane and cyclohexane. Solvents using double or triple bonds or heavy atoms are generally avoided because of the absorbance of a sample will be proportional to its molar concentration in the sample cuvette. The corrected absorption value known as the molar absorptivity is used when comparing the spectra of different compounds. This is defined as molar absorptivity is equal to AI, where A is equal to absorbance and G is equal to sample concentration in moles per liter. I is equal to length of energy light path through the cuvette in cm. For the spectrum of the light, a solution of 0.249 mg of the unsaturated aldehyde in 95% ethanol was placed on a 1 mm cm cuvette for measurement. Using the above formula, Nm will be 250 nm. Note that the absorption exceeds into visible spectrum of the light, so it is not surprising that this compound is orange colored. Molar absorptivities may be very large for strongly absorbing compounds and very small if absorption is weak.